So Michael he runs a laboratory at the Institute of Physiology at the Czech Academy of Sciences, and he has a talk about rRNA in rat metabolism. If everything's working, there we go. All right, that's working fine, Michael. So go ahead.
Okay, so hello to everybody. This is the title of my presentation. Common naturally occurring variants of mitochondrial DNA has been fixed in large population. Ancient migrations of women can be reconstructed by sequencing mitochondrial DNA from indigenous populations. Haplogroups are regional because they were founded by adaptive functional variants. This slide shows the first report of inherited mitochondrial disease mutation 25 years ago. Over time, hundreds of clinically relevant mitochondrial mutations have been identified. There are 3 clinically relevant classes of mitochondrial DNA mutation, recent deleterious mutations that results in matrilineal disease, ancient adaptive variants that likely predispose to common diseases, and somatic mutations that accumulate in tissues with age. Mitochondrial oxidative phosphorylation can be perturbed by nuclear DNA genetic alterations and/or by mitochondrial DNA mutations. Alterations in mitochondrial structure and functions can impair hotspots, which in turn can reduce energy production, alter cellular redox state, and increase productions of reactive oxidative species. Aberrant mitochondrial energetic metabolism leads to metabolic regulation, endocrine dysfunction, and symptoms such as diabetes, obesity, and cardiovascular disease. This slide shows derivation of SHR complastic strains. Since the mitochondrial DNA is inherited maternally, female offsprings are always crossed to males of the SHR recipient strain in each across generation to ensure transfer of the donor strain mitochondrial DNA onto the SHR nucleogenetic. The table summarizes amino acid differences in mitochondrial encoded oxos protein subunits present in complex strains. Mitochondrial DNA of brown Norway and origin differ from SHR mitochondrial DNA 15 and 11 amino acids. And in addition, transfer RNAs and both 12S and 16S ribosomal RNA are also polymorphic. This slide shows experimental design. SHR and SHR complastic strains were after weaning fat, standard, or high-fat diet. At week 18, the oral glucose tolerance test and the animals were sacrificed and tissues collected for analysis. High-fat diet promotes insulin resistance in complex. That can be seen in all strains. High-fat diet led to increase in body weight relative to the SHR strain. In which complex strain, the body weight was increased on either of the diets. As expected, the area under the curve for glucose during the oral glucose tolerance test higher on high-fat diet compared to controls in all groups, indicating a lower ability to clear glucose from the blood. Also, insulin levels were increased significantly 30 minutes glucose gavage on high-fat diet. Insulin concentration was significantly higher in Fischer complex present compared to the SHR. Indicates insulin. To determine whether insulin signaling is modulated by accumulation of bioactive lipids such as histamine or glycerol, from lipidomic profiling in plasma, liver, and heart in all means on high-fat diet. We did not find any effects of mitochondrial DNA haplotype on the level of amylase. However, future complex diacylglycerol were elevated in liver and heart. These results suggest that diacylglycerol are modulator of insulin signaling in Fischer-complastic rats. We hypothesize that diacylglycerol accumulation may arise from defective mitochondrial fatty acid oxidation. Therefore, focus on mitochondrial function and measure mitochondrial respiration. Can be seen there was significant decrease in the acid-linked oxphos capacity in the heart of Fischer-complastic. This graph shows the analysis of protein levels in the liver and heart in Fischer-complastic strains compared to the lower oxidative capacity in the heart of high-fat diet is caused mainly by decreased content of oxphos complex. These results indicate that the lower respiratory capacity observed in the heart of Fischer-complastic rats on high-fat diet is caused by reduced level of oxphos protein. To further test this possibility, we perform in vivo, actually it is ex vivo, experiment radioactively labeled methionine in rat skin fibroblasts derived from complastic and HRS to assess the efficiency of mitochondria. As can be seen, the amount of newly translated mitochondrial encoded protein was significantly decreased in complastic animals. This affected all evaluated, which indicates generalized drop in mitochondrial protein. The Fischer variant of mitochondrial RNR1 gene, which codes for mitochondrial encoding 12S ribosomal RNA, harbors two single nucleotide polymorphism localized in the microribosomal subunit, which go during assembly of mitochondria. So, in conclusion, naturally occurring sequence differences in mitochondrial ribosomal DNA reduce amount of proteins of mitochondrial small ribosomal subunit and slower translation in mitochondria. Under the stress condition, such as high-fat diet, lower levels of mitochondrial coded DNA, mitochondrial DNA encoded oxphos subunits result in impaired oxidative capacity. Lower oxidative capacity induces accumulation of bioactive acylglycerols in tissue and naturally occurring variants of mitochondrial ribosomal RNA may represent a risk for the development of insulin resistance. I would like to my collaborator from laboratory, from my laboratory in genetic pathology,